Okay, we're going to look at the endocrine system now. Keep in mind that um, we mentioned the idea of homeostasis a little bit already, but this is the maintenance of internal conditions inside your cell. And your body and your cells have ways of maintaining um, this maintenance through various self-regulating mechanisms, which we'll talk more about later. Um, This is very tied to your circulatory system, and when we talk about diseases, we're talking about your cells or your body being out of homeostasis. So, specifically, we're going to be looking at the endocrine system of vertebrates now. And there are animal cells, since we're focusing on humans, have the ability to communicate with one another over various types of ranges, from short to long, using chemical signals. Animal cells can communicate with adjacent cells through gap junctions, and this is when you've got two cells side by side and they can just send out the chemical from one cell directly into the next cell. And also by sending out molecules that will bind to receptors in or on other cells. So the hormones we're going to be focusing on in the endocrine system are going to be excreted by glands and other organs that produce hormones. So you might have a situation where you get a signal to your brain and it tells your hypothalamus to communicate with your communication devices, if you want to call it that way, either through the nervous system by using the neurosecretory cells that will secrete hormones into the bloodstream, or through the pituitary, which will make hormones, which will be also going into your blood vessels, into the bloodstream to move around the body. So we'll be focusing on these signals and how they are going to be responded to and received throughout the body. It is in the capillaries of your circulatory system where animal hormones get secreted from cells directly into the interstitial fluid and then picked up by the capillaries and distributed throughout the body. And of course, at other capillaries where they will be diffused out of to communicate with those parts of the body that it needs to. The endocrine system is found in lots of different parts of the body. There are hormones that we're going to be focusing on that are produced through ductless glands, meaning they don't have like an actual hole for the hormones to come out of. Um, and there are molecular receptors that will receive these hormones either on or in target cells where the response is needed. Now, animal hormones are just one type of signaling molecule that can be used. We're going to see some others here, but we're going to be focusing on animal hormones the rest of the time. But I do want to mention that if you were to put these four in order in terms of distance, neurotransmitters are going to be working shortest range from one cell to the next cell. Local signaling molecules work with a region of the body, and then animal hormones work throughout the entire body. And of course, this does leave you with pheromones, where pheromones are uh, communication hormones, chemicals, where one organism, such as one human, is able to communicate with another organism, sometimes other humans or even other organisms sometimes. This depends on the species that we're looking at in question. Bees do this as a way to communicate between the queen and the rest of the hive. Humans do also communicate through pheromones, even though we don't necessarily recognize it. A lot of times it's by what we would call scent. We smell things about other people, which is a type of communication. Now, hormones are involved in a cell-to-cell communication. Hormones or other signaling molecules end up being received at a protein receptor. And if you remember about enzymes, enzymes have these active sites where the substrate binds to the enzyme, but only specifically certain substrates can bind because of the shape of that active site. The same thing with our protein receptors. They have specific binding sites that are shape-based so that you have specific things like this square bound to the receptor that's also a square. And of course, in this case, it's bound on the outside of the cell, whereas the triangles are binding and being received on the inside of the cell, so that later, once they're bound, they will create a response from that cell, which will produce whatever that signal, that hormone is trying to get from that cell. So you can get secreting cells that will secrete certain hormones. They will only be received by a target cell. And that target cell must have a receptor of the right kind. It can be nearby, or it can be put into the bloodstream and received somewhere else in the body by cells that have those specific receptors. Sometimes hormones are bound on the inside of the cell, and you'll see this hormone receptor complex that is created, which will then sometimes go into the nucleus, which of course creates the ability for this to control what DNA is uh, transcribed and then later translated, so it controls the protein making mechanism of the cell. So a lot of times what happens is you might get some external um, stimulus where cold exposure tells your hypothalamus then to, hey, my body's cold, do something about it. It will talk to the pituitary gland, which will then talk to the thyroid, which will then talk to which will talk to the rest of the body. And in each of these steps is a hormone that's produced to talk to the next or communicate with the next thing in the series. And once we get this thyroid hormone produced, such as thyroxine, we see um, this will go throughout the rest of the body and tell the body to deal with being cold, such as start to shiver or start to contract the blood vessels at the outside of the body to retain body heat. Now there's multiple categories of hormones. There are the steroids, amines, peptides, and proteins. But technically we'll lump these into two groups, the steroids and everything else. The everything else can be referred to as either peptide or protein hormones. And the reason why we can lump them together is because they're just different sizes of the same kind of structure. Amines, peptides, proteins, in that order, amines smallest, peptides shortest, proteins largest. These three are all made up of some kind of amino acid sequence, just like all proteins are. So these are all basically protein based, and just based on size is the difference. So they're going to function the same way just because they're made of the same kind of molecules. And so we can look at hormones as just chemical messengers that will operate in very small quantities. And these small quantities is a good thing because our body doesn't need to be making lots and lots of hormones to get a response. A small amount that will give a large response is much more efficient. It's also much more quick to get a response. When a small amount produced will give the body just enough time to do what it needs to do, whereas instead of taking hours to make a big amount of a hormone to get a response, making a few molecules is enough to get the body to do what it needs to do. And so then it's secreted into the bloodstream by our ductless glands, and they bind to their receptors in the kind of lock and key style model that we've kind of seen with our enzymes. So peptide proteins are excreted out of the cell, and they're received on the outside of the actual target cell. Steroid hormones are excreted and then received on the inside. And we're going to talk about steroid hormones here first because they're made of a cholesterol molecule. So they're basically lipid based. And because of this, and the membrane being lipid based as well, the steroid hormone can just slide right through into the actual cell. So it diffuses across the plasma membrane. It creates a hormone receptor complex that's able to bind onto the promoter of a gene inside the nucleus, and then we can start gene expression of a specific gene that this binds to. So we see it coming out of the blood vessel, sliding through because it's able to diffuse across the membrane because it's made of the same kind of structure. Even into the nuclear membrane, we cause it to bind with the receptor, so you get a hormone receptor complex, and this creates something called a transcription factor that's able to bind to that promoter of the DNA and start transcription of some protein. Of course, that goes through RNA and then becomes protein after translation. Now, some examples of this are estrogen and testosterone. Now, the large amine, peptide, and protein hormones bind to a receptor of the plasma membrane. This means that they cannot go into the cell. And what happens then is they bind to the cell, and then a secondary messenger on the inside relays the message to especially the nucleus. So a lot of times the secondary messenger might be ATP, And so ATP gets converted to cyclic AMP. And if you've noticed that ATP has triphosphate, AMP is monophosphate, so we're breaking off two of our phosphates, and we create the cyclic AMP, which will activate a bunch of series of reactions. So examples are insulin and glucagon. Because they're water soluble, they're not able to pass across the membrane, which is of course hydrophobic, and then anything water soluble is hydrophilic. So it has to get bound to a receptor. And what does is it creates a signal transduction pathway where the hormone is bound to the receptor. And on the inside, that secondary messenger ATP gets turned into cyclic AMP, which will then go and cause a bunch of other series of reactions to occur. So what happens if you don't have a secondary messenger on the inside? Well, then the response won't happen. Think of it, for example, you walk up to one of these secret hideout places, and you knock on the door, and they slide the little eye slide open, and they say, "Hey, what do you want?" Well, of course, you can't get in, but as long as you have the password, they'll open the little thingy to see who you are, and you can pass a message to the secondary messenger on the inside. That secondary messenger then can go and pass the message on, so on and so forth, on to the boss who's in the nucleus. Now, what happens if no one answers the door because there's no secondary messenger on the inside? Even if you're there and you've got the password, just like these hormones are able to bind to the receptor, if there is no secondary messenger, then you won't get the message passed. So just like if there's no guy on the inside, you can stand there knocking all day long, and you've got the right password, you still won't pass your message on. So hormones can be maintained or maintaining homeostasis, and there's a couple or three ways actually to do this. There's the negative feedback loop, the positive feedback loop, and the antagonistic hormones. We're going to look at these in more detail. So remember biochemical pathways? We talked about this in Biology 1. This is where you have a substrate, use an enzyme to help this reaction take place, and you get a product, and that product creates another product, or that product becomes a substrate which creates another product, and so on. You get a series of reactions. And sometimes that end product can actually interfere. When you make too much of the end product, it will come over here and bind to the enzyme, turning off or inhibiting that enzyme, which means that we don't make this product anymore because this cannot become that if the enzyme is not there to help. And so this shuts down the entire pathway. Now, when you run out of the product, it stops interfering with the enzyme, and we can start the process of taking this substrate and turning it into that product again. This is the idea of a negative feedback loop. We see this with the hypothalamus. Let's say that your thyroid level gets really low. And so your thyroid level, if it gets really low, hypothalamus says, hey, I got to do something about it. It will make something called TRH, which talks to the pituitary, which will make TSH, which will talk to the thyroid. And the thyroid will make T3 and T4, which are what are going to move throughout the entire body to be able to cause a response. If your body's too cold, it will tell the body how to stay warm. Now, when you run out of T3, T4, this process keeps going. Now, when you start to build up too much T3 and T4, it actually goes back and stops the hypothalamus from making more TRH, which of course means you won't get TSH, which means you'll stop making T3, T4, and eventually the levels will go down, which will cause this process to start again. Okay, so as long as you have too much T3, T4, it's actually going to stop making this process. Now, a positive feedback loop is a little different. Here, instead of having the absence of something causing more growth, more production, here it's a positive stimulus where either the baby's head pushing on the cervix causes oxytocin to be produced, which causes the birth to start to take place. Or in the case of lactation, when the mother sees the baby, her mind is focused on the baby, and the baby is also suckling on the breast, this causes the oxytocin to be produced, which will cause milk to be released. So this is an outside positive stimulus. Once the outside stimulus from either the baby here or the baby there goes away, then eventually the oxytocin decreases, stops being made, and then lactation or delivery stops. An antagonistic hormone situation is a little different. This one's kind of cool because it's a balance. Let's say you have a balanced blood glucose level, but you eat something. It brings your glucose level up. Your pancreas makes insulin to bring that down, which in the body, insulin tells your body to remove the blood glucose down to more manageable levels. It can basically just use up the glucose because we see it used in cellular respiration. And it just keeps going like this until your blood glucose level goes down. Maybe you stop eating and it's been a while since you've eaten, and then your pancreas will create glucagon, which will then cause the blood glucose levels to rise. And when they rise too high, insulin is produced, and when they go too low, glucagon is produced. If it doesn't go too high, then we just keep doing glucagon until we go too high, and then we create insulin. If we stay high, and you see around and around this goes, and it's a balance of each other between insulin and glucagon. So how do we know if a cell will respond to a hormone? Cells will not respond at all to a hormone unless they have the receptor for it. One or more hormones may inhibit, enhance, or prime a target cell capacity to respond to another kind of hormone. So we saw that with the negative feedback loop, where the end product was able to actually turn off the first product to be made. Now you can also use another hormones to cause the production of things, or to help, in this case prime, a target cell to be able to be more receptive. You get different feedback mechanisms like we've just talked about, and that will help to alter different hormone concentrations, which will of course alter the cellular response. And even with enough receptors for a hormone, the certain components of the cell cytoplasm, like the lack of having ATP inside, might actually cause an effect. So if it's not available, then you won't be able to carry out the response. And then of course you also have environmental cues that mediate timing and the extent of how many hormones are secreted.